Thank you everyone for joining. The title of the talk today is The Good, the Bad, and the Smelly, the study of microbial ecology in the sediment. I'm presenting here with two of my PhD students, they're PhD candidates. Rachel Weiss. We're going to on some of our coastal work, but also some of our deep subsurface or offshore work as well. And anyway, so I'll introduce them as their talks or as they present their portion of the talk. But first, I'd like to thank several organizations and people within those organizations that have helped make this research possible. In addition to Rachel and Megan that are presenting today, we have other lab members that have been involved in this group, Morgan Sobel, Christian Cunningham, and Clay Clarkson. There are some master students that have been integral in this research, and also a former lab member, now Dr. Laura Zinke. She graduated with her PhD a couple of years ago and is now at Nature Geosciences as an editor. And folks from the Center for Dark Energy Biosphere Investigations, that's what CW stands for. It's an NSF Science and Technology Center that our group is involved with, and I have been since its inception. The Deep Carbon Observatory, especially the Deep Life community, for a lot of the biogeography work that we're going to be showing today. And of course, a lot of this work wouldn't be possible without the International Ocean Discovery Program, or IODP, and it's the Scientific Ocean Drilling Program, and then some collaborators at University of Bremen, of course. There we go. All right, so we know a lot about microbes here recently. Of course, it's been all over the news, especially the virus kind. But our idea of microbes, bacteria in particular, are that they're dangerous, that they're harmful, that we need to eradicate them all. But in reality, we do have, of course, pathogenic microbes that cause infections, can cause diseases. But we also have the most of the world, our Earth, is made up of microbes that are important ecologically. They're important in the nutrient cycling, biogeochemical cycling. They're important in our natural fauna within our guts, help us digest, and of course, other mucous membranes. And then bioremediation, we can harness their ability to attack and eat, different substrates in a way that we can use that for remediating the environment. So this is our view of life here, on land. It's relatively warm, about temperatures that are conducive that we think of life. And of course, there's light, there's sunlight, oxic. And this is where the majority of research happens, is, in these types of environments. When in reality, most of the globe is cold. It's, if we're talking about the deep ocean, it's 4 degrees Celsius, or, in the deep subsurface. And then dark, and of course, suboxic as well. So this is one drop of seawater. It's all these cells are lit up by, through DAPI staining, and you can see the larger cells being either bacteria or archaea, and the smaller cells being, or the smaller particles that you can see are viral particles. And I like this image because it reminds me of the night sky, that there are, it's important to note that, just within our ocean, there are more cells in the marine environment than there are stars in the sky. And we know vastly little about ocean and the abundance of microorganisms, but also the, who they are and of course what they're doing. And we can't plate those. We can't put them on to culture them all. We can't just take them from the environment and replicate the conditions in the lab, although we'd love to be able to do that. But, estimates are anywhere from 1 to maybe 10% of that can be grown in the lab. So what do we do instead? We use genetics. Particularly genes like the 16S ribosomal RNA gene, but also all the genes together in either metagenomics or metatranscriptomics. And we'll talk about both of those today. So this is the tree of life that you're probably very familiar with. This is bacteria in the top left, and of course, archaea in the top right. And there you are, Homo sapiens, more closely related to corn, which is Zea there, or Copernus, which is a slime mold, than one microbe or one bacteria is to another bacteria. And certainly bacteria to archaea are two different domains altogether. And if you look at the new tree of life, published just a couple of years ago, or 4 years ago now, this is, again, here's our eukaryotes in the bottom right, and our archaea is just above that, almost branching off with the eukaryotes, almost looking like that, more closely related. Archaea and eukaryotes are more closely related than, of course, bacteria are to each other or to them or to the archaea itself. But just take a look at this huge tree of life and all of the organisms that are, have vastly different functions from each other. The cell structures are completely different than each other. So really, just lumping bacteria together as one unit or as one organism is not giving them the justice that they deserve. And all the red dots that you can see here have no cultured representatives. So we use genetics and DNA and RNA to interrogate these a little bit more closely. So in our lab, we do have two ways to attack, and this is the techniques that are used with my lab group. We do a cultivation independent approach in which we both isolate DNA and RNA here on the left. We do a lot of sequencing, high throughput sequencing of both the metagenome and the metatranscriptome, and as well as the genome if we've grown them as well. And then, of course, we take those genes back to assemble and annotate them to understand more and also who they are. We also have a cultivation dependent approach as well, in which we sort microorganisms into plates. We use a live dead stain to differentiate who is active and also what cells have been damaged and are dead. And we sort it into specially designed media. And then, of course, from there, we can grow it up further and do some ecophysiology, also doing some genomic work and to do some compare those genomes to other known genomes. And we've already, just using this technique, have found about a half a dozen and maybe even more that are unique species and definitely unique strains. And we have both RNA and DNA that we work with. I've mentioned a lot about extracting both the RNA and the DNA and doing the metagenome or metatranscriptome. Now, it's important to recognize the differences between the RNA and the DNA. And of course, the DNA is the complete genome. It is it's like the library of everything that is potentially available to that cell in terms of its genetics. So it's everything that is that is possible that cell can do. It doesn't necessarily mean that the cell is doing that actual activity. So the DNA tells us, like I said, the potential of that organism. It also is important to note that the DNA can survive or can exist after the cell is dead. So you can have DNA that is extracted from a dead cell, from an inactive cell. Maybe the cell is dormant. Maybe it's a spore and it's in a dormant phase. And you can also have it, DNA that survives extracellularly. So using a DNA-based approach is good to understand the potential of the community, but it does fall short of telling us what is actually happening at the time of sampling. And that's why we also use RNA, and we rely most heavily on RNA in my lab group. So RNA is a very short-lived molecule. It's on the order of seconds to minutes from the time that the cell is, lysed or opened during extraction. So we know that if we detect RNA, that it is a proxy for activity. All right, so we're going to break this down into two categories. So our deep subsurface that you can see here, first of all, that's what Megan's going to be talking about with us today, Megan Mullis. And she's going to present on her work in this very nutrient poor environment, very carbon limited environment. A lot of the carbon is recalcitrant carbon, very little of organic carbon, and very energy limited. And in particular, she's going to talk to us about some mud volcanoes near the Mariana Trench. And Rachel's going to bring us on home with some of the coastal sediment where the carbon is more labile, it's more nutrient rich. She has about, in her samples, about 100 times to sometimes 1,000 times more carbon than the samples that Megan's working with in the deep subsurface. Another thing to note is the terminal electron acceptors that that you can see here on the bottom in this, the colorful areas where we've got the abyss, the slope, and the shelf, below this cross section. This, this is happening when you're talking about the deep subsurface. This is happening on the order of hundreds of meters into the sediment. So your activity is quite a bit slower. You're also going through those terminal electron acceptors on a much longer scale, deeper scale. Whereas the activity that Rachel is working with here on the coastal shelf, all of that can happen in the top few centimeters, and she'll talk to you more about that, so that you go through all of your terminal electron acceptors largely within the top meter, sometimes a couple of meters, but it's just happens on a much quicker scale. And with that, just to introduce a little bit, Megan will also talk to us about why, the microbial community matters and how they interact with each other in terms of their competition. And so this is just a little preview, but she'll be talking to you about antibiotics that are produced in the deep subsurface. And it's a very interesting part of the work that we're doing in my lab. And both Megan and Rachel will also talk about the methane production that is happening within their sites. And of course, the recycling of that methane, because of course it's produced by methanogens, but it's also recycled by methanotrophs. And I'll leave you with this, that the deep biosphere is alive, it is active. We're going to talk about, top few hundred meters, but it actually extends to at least 2,400 meters below seafloor. And to be fair, this is DNA based by Fumio Inagaki, this study, and Yuki Murano, but there's no, one of the things that we're trying to assess as the deep biosphere community is how life, what are the limits to this life in terms of both depth, but also in terms of the carbon utilization and the carbon availability. Okay. And Megan, take it away.
All righty. Thank you, Brandi. Again, my name is Megan Mullis, and I'll talk a lot about the deep subsurface today. But before I get into that, I wanted to talk briefly about what microorganisms need to survive within their natural environments. And first and foremost, that would be the biogenic elements, including carbon, hydrogen, nitrogen, oxygen, phosphorus, and sulfur. They also need energy to feed and support life. So they need electron donors and acceptors to perform their metabolic functions. And they also need access to water. Some microorganisms can also have interactions with each other to help them survive. And those interactions can include adaptation to their surrounding environment and their surrounding community. They can form mutualistic relationships where they can create different metabolic compounds and share those amongst each other to survive. And then they also can compete. And there's many types of competition. The three that I have listed here are you can outcompete your surrounding cells. The microbes could inhibit the growth of the surrounding cells, and they could even go as far to lyse or kill the surrounding cells and use the remaining resources for energy, or use the cell that they lysed for energy. And we're very familiar with that idea of killing or inhibiting the neighbor because of antibiotics. And as Brandi mentioned earlier with COVID, we're all very familiar because of how COVID has affected our nation, that we all need to take antibiotics to inhibit harmful bacteria. But this also happens in natural environments as well. And this little schematic here is illustrating a producing microbe, a sensitive microbe, and a resistant microbe to an antibiotic. And so the producing microbe is killing the sensitive strain because it has access to make this toxin or this antibiotic. And then that sensitive microbe doesn't expend energy to create the compound or resist the compound. The resistant bacteria produces a resistance gene, so that's taking some of its energy to resist that toxin and withstand its surroundings. And that producing microbe is producing a compound that's energy taxing. So not only are we concerned with how this ecological loop is occurring, but also how that energetic expenditure is happening within all these natural environments, especially in deep subsurface when the energy is so limited. Are they going to expend that extra amount of energy to produce these antibiotics? And so I have this gene tree for this antibiotic from the non-ribosomal peptide synthase gene. And you can see these stars are highlighting different branches of this tree where deep subsurface samples are located. And these are from the western flank of the Mid-Atlantic Ridge and also Juan de Fuca Ridge. And so these are basaltic fluids where we analyzed the metagenome and found some of these common antibiotic genes. And they were similar to our terrestrial analogs, but they're still very different. So what we want to know is how they're different and what energy they're using to make that compound, if they are making it. And this is another tree for another common antibiotic gene, the polyketide synthase. And the polyketide synthase is very similar to some other genes for cell components. So typically you will have more genes show up in your metagenome for this gene in particular. So that's why this tree is a bit bigger. And again, these stars are signifying the different deep subsurface data sets that have appeared in this tree. And again, they're very different than our terrestrial analogs. So not only is this, a medical question, but also very much an environmental question, because we're not totally sure how these microbes are creating these compounds and for what purpose. A little bit more about the marine sediments, especially deep subsurface. As you move further and further away from land, the cell count drastically decreases. So you can see in this panel C here, this global picture with the red hue surrounding the continents. That's showing how the cell count is very high. And you can see the scale bar below it. And as you move further and further away from the land, you get less and less cell count, and you can see the log cell concentration in panel A, where those yellow dots are from the South Pacific Gyre. And the South Pacific Gyre is the furthest point from any landmass, and it's deemed the ocean desert for that reason. It doesn't receive a lot of input. It has very recalcitrant carbon, very energy limited, and low cellular abundance. So it is characterized as an extreme environment. The Mariana Forearc is very similar in being extreme, but it has different extreme nature than South Pacific Gyre. This is a simple schematic of the Mariana system on the bottom left. You can see the trenches in purple, and then the forearc, which I'll be talking about, the volcanic arc, and the back arc, and the remnant arc. In the bottom right figure is the IODP cruise expedition 366 that went to the Mariana forearc and retrieved these samples. So thank you again to the science party and crew for collecting those. This is a more clear picture of where the samples were collected. They're from 3 different seamounts, Yenazao, Fantagisnia, and Asut Tesoru seamounts. And each of these seamounts were sampled at the summit or the top of that mud volcano and the flank of that mud volcano at Asut Tesoru. So the flanks were sampled at Asut Tesoru for this study as well. And this is a horizontal schematic of that forearc system. So you can see the Pacific Plate seamount is subducting under the Philippine Plate, which causes these forearc mud volcanoes to form. And these orange stars are highlighting where these mud volcanoes are that we studied for this project. And something that's unique about these seamounts is that they're serpentinizing seamounts. In a very basic definition, serpentinization happens when mantle rock below the sediment is lifted up and interacts with the seawater above. And upon that interaction, there is a massive release of hydrogen, and it can cause a higher pH, and it can also cause methane to form. And a good indicator of serpentinization happening is the color of these sediments, and that's why I put this figure on here from the expedition. This is a cross section of the sediments collected from the Mariana forearc, and when serpentinization happens, you can see there's a blue-green tint to the sediments, and that's a really good indicator that this is occurring or has occurred. And so something that's really important from this serpentinization reaction is it could lead to hydrogen metabolisms, methanogenesis, and methanotrophy. And so this is a depth profile of the geochemistry collected on board. It's selective geochemistry. There was much more taken, but what we're interested in is the sulfate, methane, and pH. And so very simply, you can see on the y-axis, it increases with depth, and on the x-axis is the concentrations of each of these indicators. And so you can see that in Yenazao and Asut Tesoru, sulfate was very high relative to Fantagisnia. Methane was extremely high in Asut Tesoru, and the pH was very high in Yenazao and Asut Tesoru. Asut Tesoru even made it to 12.5, and we know that that is the limit to life for pH as of now. So that's an excellent indicator to how life can persist in this extreme environment. So our objective was to look at the RNA or the metatranscriptome of the system, and we wanted to identify and quantify transcripts for metabolism and antibiotic production. And we hypothesized that methanogenesis and methanotrophy, or the making and consumption of methane, would be highly abundant relative to other functions. And we also thought that antibiotic gene expression would be upregulated with respect to primary metabolic functions. So just a basic overview of the microbial community. We had archaea, bacteria, and eukaryotes found within the sample. And they, again, this is RNA, so we think that these are active members of the community. And we saw very little archaea, pretty diverse bacterial community, and it was pretty similar across the board. And interestingly, we saw a Piscicantha from eukaryotes, which comprises a lot of fungi, and fungi are pretty hardy. So we suspect that that could be why they are withstanding this environment, is because they could withstand extreme environments. And panel B is a simple dendrogram illustrating how similar or dissimilar these samples are to one another. And the triangles indicate summit sites and the squares indicate flank sites. And the two things I want to take away here is that the summit site in purple, that's from the same seamount, the same summit. It's just different depths. So those are understandably similar to each other because it's the same site. And then the flank sites are most similar to each other, which is very comforting to know that the flanks are different than the summits, which would make sense because the flanks have been out of that serpentinizing reaction a little bit longer than the summit sites. And for the bottom panel in panel C, we have this non-metric multidimensional analysis plot, and we have this these two arrows are indicating the flank sites from Asutasuru, and the main point here is that the flanks again are different than the summit sites, and we found the significant drivers of this community diversity to be calcium, strontium, depth, and sulfate. For the functions, we saw most of the functionality was for metabolic process and catalytic activity, and there was a good amount for unclassified as well. Within the unclassified, there's a lot of hypothetical proteins that we cannot actively state what function it is, so that's why they're in that unclassified category. And the main metabolisms that we saw are highlighted here with these stars. We saw an overwhelming amount of gene expression for anaerobic methane oxidation and partial denitrification, especially from nitrite to nitric oxide and nitrous oxide to dinitrogen. And we see that across the board for all of those samples. So we think that there's some coupling of those reactions to make the energy expenditure less for those cells and the payoff more. And we did a canonical correspondence analysis of these metabolisms and the geochemistry. And we see here the black text is showing all of the sites. And again, all of the summits clustered together while the flanks were distinctly different. And in this case, for the metabolisms, we saw iron, silica, magnesium, lithium, and bromide to be the significant drivers of gene expression. And finally, a previous study done on the Mariana back arc from Trimbeth Rakard et al. In 2019 found these various metabolisms within their metatranscriptome data. And the main difference that I wanted to draw attention to here is that the Mariana forearc has much higher gene expression. And also, again, we see that methane oxidation and denitrification showing up in our transcriptome data set. And we also see a good amount of hydrogen utilization as well, but not to the extent of methane oxidation and denitrification. And so from that study, we can further investigate how these microbes are withstanding this extreme environment of the deep subsurface.
And I was going to go ahead and introduce Rachel's portion. And again, she's like Megan, a fifth-year PhD candidate, both getting ready to graduate in the very near future. But Megan just talked to us about the deep subsurface, and so we're going to bring it closer to home with the coastal sediment that is more labile, more carbon-rich, more nutrient-rich. And again, those activities that Megan talked about are happening on a much shorter, both in terms of distance and in terms of time scale.
Okay. So as Brandi just mentioned, I'm going to be talking more about coastal sediments. And one of the fun things about this project is we really get to focus on how the microbial community is driving this geochemistry, with the added bonus of looking at some vegetation that is also in our sediments. So we're really going to be in this sweet spot here in the middle of all of those circles, looking at microbial communities, vegetation, and geochemistry. And for part of the reason that we're looking at this is because we're really interested in that carbon cycle, that methane production. And when looking at different types of vegetation within wetland systems, we often hear about seagrass, spartina, and mangrove. And we're really interested in how carbon is stored in those systems. And I want to point out with this slide that carbon is stored the most in our mangrove systems, but mangroves usually take up the least amount of area. So we're really interested to see with mangrove encroachment, as mangroves expand upward into past northern limits, as well as into other previously established vegetation types, how is this going to affect our carbon stock? So really looking into how is this going to change the microbial communities, as well as that biogeochemical cycling? Are we going to have more methane production? Are we going to have more emissions? And part of the reason for that is because about 736 tetragrams of methane are emitted into the atmosphere annually, and wetlands are producing about 164 of that. And from that, 80% is produced by biogenic methanogenesis. So that's that microbial community really putting in the work and producing a lot of methane. And we have to think about when methane's being produced, we have to think about methane being consumed. So I'm going to be talking about this little loop on the far left, this methanotrophy cycle, where when we have a methane value or methane flux value in our site, we have to remember that that's not necessarily indicative of all of the methane being produced, because there is a chance that some of that is being consumed by methanotrophs and producing carbon dioxide instead. So we're really going to be looking into our total and active microbial communities and really piecing out this methanotrophy, methanogenesis co-occurrence. So we were at Mission Aransas National Estuarine Research Reserve, which is in South Texas. And we went to 3 different vegetation sites, a seagrass site, Spartina site, and mangrove site, as shown on that map there. And we went 3 times, and we also went day and night because we just love going to the field. So one of the things that I want to point out with that day and night sampling is that we actually did see some diurnal variations. Just to orient you, what I'm showing here is concentrations of sulfide at the y-axis or the x-axis and depth on the y-axis. And we're moving throughout the sediment column, and each row is a different series, different time that we went sampling, with our different colors responding to different vegetation types. And the stars are signifying that there was a statistically significant difference between our daytime and nighttime sampling. Mainly wanted to point out here that with our mangrove sampling events, we happened to see much, there was more statistically significant differences, whereas with Spartina and seagrass, it was a little more variable. Another thing I just want to point out is that in our November sampling event, we had much higher ranges in sulfide concentration. So thinking about how diurnal variations are being seen in our sulfide concentrations, let's take a look at how they're looking for methane flux. So this is a calculated methane flux from discrete measurements of concentrations of methane throughout the upper 6 centimeters of our sediment column. And so what we're showing here is these negative numbers are the amount of micromoles of methane per centimeter squared per day being fluxed into the atmosphere from those 6 centimeters of the sediment column. One thing I wanted to point out from the get-go is that in our seagrass samples, they were consistently low. There wasn't much change from day to night, and there wasn't a lot of variability from sampling event to sampling event. However, in our June sampling events for both mangrove and Spartina dominated sediments, we saw that there was higher fluxes of methane during the day. And then inversely, in August, we saw the opposite trend, that there were higher concentrations of methane during the nighttime event. And again, we're talking about the top 6 centimeters here. So part of the reason that we could calculate this is because we took a depth profile of methane concentrations throughout our sediment cores. And this will come into handy when we start talking about microbial communities at these 0 to 12 and 20 centimeter depths. And then another way that we looked at methane flux was by deploying flux chambers, which is what this bottom right image is showing here in a mangrove field. And what we calculated here was for each one of these replicates, how did methane flux into the atmosphere? And what I want to note is that these are on different scales, but we did see the same trends, where at the nighttime for August sampling events, we had higher concentrations of methane, especially in our Spartina-dominated sediment. So when we're looking at those 2, 12, and 20 centimeter depths, this is actually what the microbial communities look like. I know this is a lot to look at, don't really worry about it too much, but what we're looking at here is 55 metagenomes that we pulled the community analysis from. And we're looking at this at the family level with way more bacteria being present than our archaea counterparts. And there's a few differences that we can kind of see without really going too into depth. This first thing that I'm pointing out is the difference with the water column. Thankfully, water does have a different bacterial community than the sediment column. And another that I'd like to point out is these deeper sediments. So while I had mentioned 2, 12, and 20 centimeters, we were able to actually go down to a range of 40 to 60 centimeters and look at the microbial communities there. Now, when we're talking about methanogens and methanotrophs, we can actually tease apart these communities and look for just those. So what I'm showing here in the pinks and reds are the methanogens, so those are producing methane, and in the blues, the methanotrophs, which are consuming it. And I have this broken up by day and night, as well as by depth and vegetation. And mainly what I want to show here is that we do see methanogens consistently throughout the sediment column, except for 2 centimeters in our Spartina samples. But we also see a lot more methanotrophs present. So we're really going to be able to start looking at what the difference between these microbial communities are and how is that going to affect their geochemical cycling. So looking back at all of those samples, this is taking a snapshot of all of the samples that we took at 2 centimeters. This is a Bray-Curtis analysis where we're looking at similarity. So the lighter squares are going to be more similar, and the top right is going to be identical to the bottom left. And mainly for this, I just want to point out that at 2 centimeters, all of our mangrove-based sediment samples are grouping together, and it's a little more variable between unvegetated, or our seagrass samples and Spartina samples. And then if we go deeper, about 12 centimeters, we're still seeing our mangrove samples really grouped together, but this time we're looking at only the November and August samples. And then as we go even deeper into the sediment column at 20 centimeters, we're not really seeing a trend here. And this can be because we're not having any effects from the overlying water column. It's a lot harder for oxygen to kind of get down into the sediment column. And we're having a little more consistency in geochemical reactions that are happening. So when we've been talking about in the previous parts of the discussion today, these metagenomes and metatranscriptomes, we've been talking a lot about what DNA and RNA are and how they're functioning. So for us, I'm just trying to show how we had 55 metagenome samples and about 62 metatranscriptome samples. And here I'm just showing they're different, which is, again, comforting that they are, in fact, different. And so we can really use this to start looking at different genes and their abundances within individual samples. So the genes that I'm really going to be focusing on today are going to be related to methanotrophs and methanogens, with methanotrophs having the gene PMO and methanogens having the gene MCR. And so really what I'm trying to drive home here is that these are co-occurring genes. So where we have methanogenesis happening, we're also having methanotrophy. And that methane that's being produced, it can escape into the atmosphere from plant-mediated transport, ebullition, kind of like champagne bubbles, general diffusion, or it can be oxidized and turned into carbon dioxide, or consumed by methanotrophs and then turned into carbon dioxide. So this is looking at those specific genes. So to orient you guys again, the x-axis is going to be our different types of genes and the pathways that they relate to. So you can see the MCRA gene and the PMO gene. And then we also have it broken up by the time that we went sampling, as well as the DNA versus RNA. So first, we're going to kind of just focus on the metagenome portion. And really what I want to show here is our variation in methane production and methane consumption, mainly for our methanogenesis, that MCRA gene. It's a little sporadic, but it's fairly present in our mangrove-dominated sediment. Whereas for our PMO gene, our methane consumption, it's really mainly in our methane-related or mangrove-related sediments. Now, if we take a look at the metatranscriptome based, which is going to be what's active and what is being transcribed, we have a lot more going on for the methanogenesis pathway. So it's a little more consistent, but still pretty prevalent in our mangrove-dominated sediments. Whereas for our methane oxidation, again, really just focusing on those mangrove-dominated sediments. So this leads us to a lot of questions about how do all of our geochemical signals that we've collected relate to our microbial communities, and how are those microbial communities driving those geochemical cycles? And so this is a PCA that we had made, taking all of our sediment samples and a bunch of geochemical signals that we had collected, and trying to see how do our sediment samples tease out. And we really saw that methane was a driver in our mangrove-dominated sediments for the daytime, which is that panel on the left there. And in the nighttime, we saw a little more variation in our Spartina-dominated samples. So what we're really excited to do for the rest of my dissertation is kind of tease apart this relationship and begin answering a little bit more of this cryptic carbon cycling related to microbial communities. And so with that, we have a lot of funding and student support I would like to thank.
Yes, thank you for both Megan and Rachel, because we're going to take some questions here. So, but yes, can't forget our funders. And then, of course, this is some nice drone footage that one of the students took, during our sampling trip to the mangroves. And the newly created lab logo that everybody contributed to, but Rachel did the artwork on. We've been very proud of that. So with that, are there any questions? You can feel free to include them in the chat box, or just jump right in there and ask the question, or certainly you can virtually raise your hand as well.
Well, if nobody goes, I have a question for Megan. First off, thank you, everybody, and welcome to Dauphin Island in South Alabama. Exciting research. Megan, I'm curious about the serpentization on that Asut Tesoro mound. The pH went up to 12, you said?
12 and a half.
Okay.
Actually, well, I'm not sharing my screen, but I can go back one second. Let me pull it up for us.
So a trick that we use in silicon and phosphorus chemistry is forming magnesium hydroxide to scavenge phosphorus and silicon at really high pHs. Mm-hmm. The serpentization reaction you showed has spruceite or magnesium hydroxide as a byproduct. Mm-hmm. Imagining that scavenges all the phosphorus and available, and maybe even the arsenate available. Mm-hmm. How on earth are things getting phosphorus in these chemical conditions? Are they just drilling it from the rock? Or, I mean, this seems like a really difficult environment to persist in if you need something to make a lipid.
That's an excellent question. From the metatranscriptome data, I used various annotation programs, and one of them is very much for assimilatory processes and cellular maintenance. And there was a lot of phosphate and phosphorus transporters in there. So I'm assuming that they would, since that expression is higher and it's actually present in these samples, that they would be scavenging for whatever phosphorus they can find. So I'm not entirely sure where it's coming from, but I do see expression for transporting phosphorus.
Can I jump in here real quick?
Sure.
Of course, there's, inorganic phosphorus that's available, but also what's available, and it's, as Megan pointed out, is the scavenging of other microorganisms. So using the other cells that are surrounding you in order to, to scavenge that phosphorus as well. And these are at the, limits of life, both in terms of the pH, but also the turnover time based on protein racemization experiments in these subsurface sites are on the order of 100 years, 1,000 years. Some have even calculated as much as 10,000 years. So we're talking a very, very, very slow lifestyle.
Just a quick follow-up to that. This might have been debunked, but I remember a couple years back that there was some work out of Mono Lake saying that, you know, arsenic could be substituted for phosphorus. Is that completely bogus in an environment like this, or is that something that
Yeah, that study
laughing.
that study unfortunately was flawed, you know, and it has not been retracted, but it was shown to be highly flawed in that the proper controls were not taken, and those organisms were in fact using phosphorus. So we still have not shown that arsenic can be substituted for phosphorus in the backbone of DNA as of yet, although it's you know, an interesting hypothesis because of both the size and the molecule itself, but unfortunately, we don't think that is plausible here on Earth yet.
Thank you, all three. Appreciate it. Wonderful presentation.
Thank you. We have a question on the chat from Blair. Wants to know if, and this is directed at you, Rachel, if you found Vibrios in your coastal sediment, and have you been able to see any relationships between the cell size and nutrient cycling? So it sounds like a little, maybe two questions there. So the first question is if you found Vibrios and if there's been any variability in those Vibrios.
I believe we have found some, but I will be candid that I haven't gone completely in depth in Vibrios. I've been really just excited to dive into methanogenesis. So I will take a look into that and double check that. But same thing goes for cell size and nutrient cycling potential. The second chapter of my dissertation is going to be really looking into the potential for different nutrient cycling. So now that we've gotten all of this data back from sequencing facility, really excited to also go into that as well. Yeah. So sorry, I don't have an answer for you, but we will soon.
In general, so the microbial cell size in the coastal sediment is larger and compared to the deep subsurface cells, there are smaller in both size and also in terms of abundance as well. So size doesn't necessarily matter when it comes to nutrient cycling, but it does mean that there can be, you know, the nutrients can be recycled more quickly, you know, on the in the coastal environment than it is on the subsurface environment, because again, you do have the carbon availability that varies so much. Do you have any other questions? Feel free to jump on in there.
I'll gladly ask another one. You guys showed a really interesting schematic about kind of the feedbacks between potentially three organisms and how, you know, the one organism creating kind of an antibiotic might facilitate another organism persisting in that environment. In the water column, we have, there's been a similar hypothesis called the Black Queen, where, you know, genome streamlining in Prochlorococcus has caused them to lose the ability to degrade peroxide. So they don't have any peroxidase enzymes, but they rely on that from leaky enzymes from other plankton. Do you guys have kind of more specific examples of that type of interaction in the sediment, or is that just kind of more theoretical at this point?
My thought from the various research in the review paper that Brandi and I and co-authors wrote, all of the producers of antibiotics or toxins in general, they tend to carry the resistance gene with them because when they produce that toxin, they don't want to infect themselves and, you know, cause them any harm. So I think based off of the Black Queen hypothesis too, that is an interesting dynamic to look at the gene evolution. And that's why we did those gene trees as well. So I think looking at that evolution of those genes over time and how it's changed, and how those resistance genes are transferred as well, that could be similar to that black-green hypothesis. And that's, from what I've read, a lot of individuals are using resistance genes as kind of a way to navigate data to see if there's any producing genes as well. So typically one can't go without the other, from what I've seen. I don't know if that answers the whole question.
There's also an energy cost to both carrying the gene and also expressing the gene, both in the terms of resistance and also in terms of production. So it's hard to say, if this is, truly being expressed and under what conditions it's being expressed.
Thank you.
Great question. All right. Do we have any other questions? Well, thank you all for attending. And oh, we've got one more. Oh, no, there was a thank you. But yeah, thank you all for attending. And we're very glad to be here and excited to, share more research with you in the future.